So I'm going to say a few words about this question. Why do some people remain cognitively able in old age? And this is something that neurologists call cognitive reserve. So I'm sure you're aware that the proportion of population in most countries that is in old age is increasing. And unfortunately, aging does bring cognitive problems which owe to brain changes. So if you can understand how aging affects the brain, we might be able to better understand how it affects cognition and come up with interventions that might help keep our brain healthy and allow people to maintain their independent lives for longer. In other words, to increase the cognitive health span to catch up with the increases in lifespan that we're all benefiting from. So a common view is that aging inevitably causes deterioration in our brains and also our cognition. But it's more likely that the effects on cognition, the effects of age on cognition are mediated by your brain health. So it's possible that other factors, such as lifestyle, may allow us to maintain our cognitive abilities. In other words, it's brain changes rather than chronological age per se that may be important for maintaining your cognition. And there's lots of different ways of measuring the brain. One way is MRI. Some of you may have seen our MRI scanner on your tour. And MRI is an amazingly flexible technology because we can measure things like brain structure, the volume of grey matter and white matter in people's brains. But we can also measure brain activity and brain connectivity, so how different brain areas correlate in their activity over time. But normally, neurologists will focus on brain structure. And this is a horizontal section through the brain of a 19-year-old. In a moment, I'll show you brains of people of increasing age up to 86. And if you watch closely, you'll see around the edge of the brain is the grey matter. That's where the neurons in our brain live. And that tends to shrink as we get older, as does the white matter. That's the slightly brighter tissue that allows neurons in one part of the brain to talk to neurons in another part of the brain. That also shrinks. As a consequence, these dark areas in the middle of the image, which are called ventricles, they expand. So these ventricles are actually just filled with cerebral spinal fluid because our whole brain sits in a bath of cerebral spinal fluid. But they increase because the grey matter and the white matter are shrinking. And if you compare the 19-year-old on the left with the 86-year-old on the right, it's quite dramatic, unfortunately, of the effects, the ravages of time on our brains. The person on the right, who's 86, is functioning very well. So this is not someone with Alzheimer's. This is a healthy 86-year-old. But it's quite scary to see this effect. If we also measure people's cognition, some things remain intact, like our language, our ability to comprehend what people are saying. But other abilities, such as what we call fluid intelligence or rapid reasoning, that declines as we get older, as does memory. And in fact, if we measure the volume of parts of the brain, such as the hippocampus, which we know is very important for memory, here we have data from 2 time points from lots of people, so each person is a line here, and you can see that everyone shows a decline in the hippocampal volume over time. But if we measure people's memory, some people do show a decline in their memory. That's quite common. But there are others who, despite the fact the hippocampus is shrinking, just like everyone else's, they don't seem to show much of a decline in their memory abilities. And this is what neurologists have observed many years ago. They call it cognitive reserve. The fact that you can take 2 people whose brain scans look very similar. So these are different brains, and the person on the left might show classic age-related problems such as slowing, word-finding difficulties, memory problems, and the person on the right may show very little sign of cognitive deficits. And you can't detect that from the structure of their brain. The brain scans look very similar. So cognitive reserve is kind of a rag-bag term that we need to try and explain away. What is it about the person on the right that allows them to maintain their cognitive abilities if it's not their brain structure? What factors affect cognitive reserve? Well, we've known for a long time that education is quite important. High levels of education lead to better cognitive abilities later in life. But in the study I'm about to show you, we are more interested in more modifiable lifestyle factors. And the study I'm going to show you comes from a cohort here called CAMCAN, which stands for the Cambridge Centre for Aging and Neuroscience, which started 15 years ago. And we recruited about 700 adults, that's about 100 per decade from 18 to 88, who came into the lab for detailed cognitive assessment and detailed brain scans using, for example, MRI. And if you want to read more about CamCan, this is the website. Some of you here may actually be CamCan members, I don't know. But using this data set, what ideally we'd like to do is track these people over 20, 30, 40 years and measure their brains, measure their cognition, and measure their lifestyle, and see how these relate to each other. But these gold standard prospective studies are very hard to do over 20 or 30 years, very expensive, and very few such studies exist. So for the experiment I'm about to show you, we had to rely on what's called a retrospective study, so that we have data about people's brains and their cognition, and we ask them to think back about earlier in their life and tell us about their lifestyles when they were a child, when they're middle-aged, and what they're currently doing. There are obviously caveats to this kind of approach, but it's the best we can do for these sort of long timescale causes. So the study I want to tell you about was done by Dennis Chan, who's a neurologist here in Cambridge. And what we did is we took 205 people in the CamCan sample who were over 65 years of age and retired. And we asked them, we gave them a questionnaire called the Lifetime Experience Questionnaire, the LEQ. We also measured their cognition using a standard test, and we measured their brain structure by the total grey matter volume in their MRI scan. This questionnaire, the LEQ, it basically gives you 6 numbers which come from 3 time periods: youth, midlife, and late life. So remember, all the people here were in their later lives, but they were recalling what they did earlier. And for each time period, there are questions that are specific to that time period. So for youth, that's largely education. For midlife, that's largely occupation. And there are other sort of questions called non-specific questions, which are repeated across the 3 time periods. So how often did you play sport, for example? How often did you see family and friends? How often did you play a musical instrument, etc.? So from these, we get a score. It's just the same thing again. So we have 6 numbers. And if you take any of those scores and correlate them with cognitive ability, there's always a positive correlation. But what we did here is what's called multiple regression. We ask which of these 6 factors make unique contributions to people's old age cognition above age and sex. And here are the results, and I'll walk you through them. We found a significant negative effect of age. So the older you were between 65 and 88, the worse your cognition, and that's normal aging. We also found a positive contribution of what the LEQ calls youth-specific activities, which is largely education. So that replicates this well-known finding that the more education you have, the better your cognitive abilities in late life. But the interesting finding was there's also a unique contribution from what people did in their midlife outside the workplace, non-specific activities. And this is, remember, this is a unique contribution above education, above their current activities. So midlife activities seem to help preserve cognition in old age, but to qualify for this concept of cognitive reserve that I told you about, they need to affect the relationship between your cognition and your brain. So as I said, we measured brain health by total grey matter, adjusted for your head size, and we asked whether these midlife activities affect the relationship between your cognitive health and your brain health. And this is the main result. It's complicated, but I'll walk you through it. So on the y-axis, we have a measure of cognitive ability, and this has just been mean corrected, so the average is 0. On the x-axis, we have a measure of your brain structural health, again, corrected to have a mean of 0. And this is the total amount of grey matter in your brain adjusted for your head size. And each point is 1 person, and I've coloured the points into 2 groups, the red and the blue. The red group are people who had lower than average midlife activities, and the blue group are people who have higher than average midlife activities. And as you can see, the red group, there was a positive relationship between their cognitive ability and their grey matter. So the more grey matter they had in old age, the better their cognition. But the blue group, those people that had higher than average midlife activities, their cognitive ability was independent of their grey matter. So something about these midlife activities made them resilient or robust to the inevitable grey matter loss that we all experience as we get older. So in other words, we've identified a type of cognitive reserve, midlife activities, stuff that you do outside the workplace, sport, family and friends, instruments, etc., that predicts cognition later in life over and above your education and over and above what you're currently doing in old age. It reduced the dependency of your cognition on your brain structure, and it's potentially easier to modify than perhaps education if we can nudge people to have healthier lifestyle choices, particularly in middle age, they might be able to preserve their cognition for longer and live independently for longer, 20 or 30 years later. So that's the hope. But you might be thinking, if it's not brain structure, there must be something about these midlife activities that affects the brain. Something else about the brain in these people is special. And here we come back to the idea that MRI can measure not just structure, but function. In particular, connectivity. Because if you put someone in the scanner at rest and measure the correlation between activity in different parts of the brain, what you'll see is a number of large-scale networks of areas that are highly correlated with each other and not correlated with areas in other networks. And then we can summarize those networks with something called functional segregation, which is how well separated those networks are. So very briefly, if we divide the brain up into different regions, that's the colors on the left, we find that the red areas all have similar activity patterns, as do the blue areas. They have similar connectivity. Or if we express it as a matrix on the right, where we have region by region, the areas within the network, the dark grey squares, are more connected than the areas outside. So this segregation measure is simply the within network connectivity minus between network connectivity. And higher segregation it means your networks are better separated. So I wanted to ask, do individuals with high levels of midlife activities have higher segregation of their networks? And does this compensate for this structural loss? And this is a paper led by Petr Rakov. We actually had 3 different states that we can measure connectivity in the CAM-CAN sample: at rest, or watching a movie, or doing a simple task. It doesn't matter what state you measure. As you get older, segregation of your networks decreases. And importantly, if we remove the effect of age, there's a positive relationship between your segregation and your cognition. So the more separated your networks, the better your cognitive ability. So that sounds very hopeful, that segregation might be this signature in the brain of healthy activities that lead to cognitive reserve. Unfortunately, we didn't find a relationship between these midlife activities and functional segregation. So our hypothesis was not confirmed. So we don't yet know what the brain correlates of this cognitive reserve are, or this midlife activities. And that's current research we're working at the moment. Is it something else? Is it functional activity, compensation, reorganization, plasticity, all things that we can measure during tasks? And does this, or is it other types of brain structure, such as white matter? And that's currently what we're doing here. So to summarize, midlife activities seem to help to maintain cognition decades later in late life. It make your cognition more robust to the inevitable loss of grey matter that we all experience. But the functional brain correlates of this robustness remain to be found. And this is the take-home message. So thank you for your attention.